Very good morning, the future of India. I am Dr. Samuel, a represent from the Department of Biotechnology of Ayya Nadar Janaki Ammal College. Beloved students, it's the time for me to acknowledge the special persons who always encouraging me to do these kind of assignments. So, first of all, I would like to thank our beloved correspondent, our beloved principal, as well as my department head for giving me an opportunity to do this assignment for the beneficiary of the students. Right, students, in this topic, in this video tutorial, we are going to see about the genetic recombination in bacteria. Bacteria are the lowermost organism in the earth. It is very primitive and it is very easy to deal with. My dear friends, and there are different methods of recombination in bacteria. The first one is transformation, second one is transduction, and third one is conjugation. So these three methods are ultimately known for to study the genetic exchange in lower organisms like bacteria. So first of all, in this video tutorial, what we are going to discuss is transformation. So transformation was first explained by Liederberg and Tatum. So who explained this topic in a great way? So according to the scientists, transformation is a process in which bacteria is able to uptake the naked DNA molecule from the medium. See, this kind of direct uptake is not taken over by almost all bacteria, and this process is very unique to certain group of population growing in a nutrient medium, and those kind of cells are said to be competent cells. They are alone able to take up the naked DNA molecule from the nutrient medium. See, the first step in transformation process is the binding of nucleic acid to the cell wall. See, bacteria containing a cell wall which is very thick in nature, the cells which are in competent state are able to express special type of proteins over the surface of cell wall. These proteins are able to attach to the DNA molecule present in the medium. So during the active process of transformation, the DNA molecule present in the media is adsorbed to the surface of the proteins which are expressed on the competent cells. Thereby, in further steps, the DNA adhere to the surface of proteins will be taken into the cell. See, those proteins expressed on the cell surface are said to be transformosome buds. Transformosome buds, they are special proteins expressed on the surface of bacterial cell wall only during the competent stage. You cannot observe the same kind of protein on the bacterial cell at all times, right? So once it is adsorbed to the cell wall, the DNA adsorbed to the protein, the cell surface protein is taken up into the bacterial cellular environment. So where the DNA gets incorporated itself into the chromosome of bacteria. See, the chromosome means you cannot see the chromosome as it is well organized in human beings, whereas in bacteria, since it is a lowermost organism, the DNA is in the form of tangled mass, which is like a floating material in the cytoplasm. See, integration of the genetic material which is coming from the different source into the bacteria is not a simple thing. This integration is carried out by a special kind of enzyme called as integrase. Integrase will help the DNA which is derived from another bacteria to incorporate itself into the chromosomal DNA of bacterium. Right. What is the use of this kind of process, this kind of complex biological phenomenon? As we all know that the DNA is the unit of heredity. Unit of heredity means it is capable of transmitting the information from one generation to another generation. Since the DNA is derived from another important bacterial cell, it may contain some usable properties, which may be metal degradation, which may be antibiotic resistance, whatever it is. The DNA which is derived from another cell, which is derived from the other cell, gets incorporated into the chromosomal DNA of the bacteria, confers the organism with new property, right? Now this organism becomes antibiotic resistance and transfer itself into a dreadful one against many diseases, right? So this is about transformation process. And the next process is said to be transduction. Transduction, it's another important genetic recombination or gene transfer mechanism in bacteria. So in case of transformation, the DNA alone transferred into the bacterial cell from the medium. Say you may ask, sir, from where this DNA has been raised. So the question, the answer for this question is the DNA is generated from another degraded cell, or as a result of death of another bacterium, the DNA may be released into the media. So the DNA is ultimately from a new organism, from the new source. Right, in case of transduction, the same kind of genetic transfer mechanism is carried out with the help of a virus. Here the viruses are called as bacteriophages. As you all know that bacteriophages are viruses feed on bacteria, means these viruses are able to kill another bacterial cell. So those kind of viruses are said to be bacteriophages. In case of transduction, and which is explained by Joshua Lederberg, and according to him, transduction is a process in which the DNA is exchanged between two bacterial cells through the virus, through the viruses. Here the viruses are called as bacteriophages, since it feed on bacteria. Fine. So bacterial viruses are able to multiply in two different ways in the suitable host cell. So one is said to be lytic cycle, another process is said to be lysogenic cycle. In lytic cycle, ultimately the host cell will be damaged, or it is completely disintegrated into many pieces, whereas in lysogenic cycle the host cell is safe, right? So let us see in detail how this lytic cycle and lysogenic cycle takes place. So in the first step of lytic cycle is adsorption of bacterial virus to the surface of bacterial cell wall. This binding is receptor mediated binding. The viruses will not bind at all places over the bacterial cell surface. It will bind to only the specific area, said to be some receptors called as teichoic acid molecules, which are expressed on the surface of cell wall, are acting as the receptor or binding sites for the bacterial viruses. So with the help of tailpins, bacterial viruses are tightly adsorbed to the surface of bacterial cell wall. Once the adsorption becomes stronger, the next step is insertion of the pilus. So the pilus is, the structure of the pilus is very similar to the hollow syringe. So the head portion of the bacterial virus containing the nucleic acid, once the adsorption becomes completed, the head of the bacterial virus becomes started to shrink. So as a result of shrinkage of virus head, the nucleic acid in the head portion of the bacterial virus gets transmitted slowly or gradually into the bacterial cell wall, right? So when the nucleic acid reaches a chemical said to be phosphatidylcholine, which is on the cell membrane of bacteria, and now that molecule becomes inserted, it becomes completely enter into the cytoplasm, right? Once it is entered, soon after the entry of nucleic acid of virus, it started to take over the complete charge of the whole bacterium. Now the nucleic acid of the virus becomes the dictator, right? Whatever the information conveyed by the viral nucleic acid, according to the information the bacteria will react. Fine. In this process, once after the entry of nucleic acid into the bacteria, this nucleic acid becomes integrated into the bacterial chromosome, and this kind of life cycle is said to be lysogenic cycle. But in lytic cycle, the viral nucleic acid started to dictate the information to the bacterial cell. So after the entry of nucleic acid, it combines with bacterial ribosomes. Ribosomes are protein synthetic machineries present in the cytoplasm of bacteria, which help the nucleic acid of virus to translate. So during translation, two different types of proteins are synthesized. One is early proteins, another one is late proteins. Late proteins are structural proteins required to build up the structure of viruses. Early proteins are enzymes required for the digestion of bacterial nucleic acid into small fragments. So with the help of these two equipments, the virus the virus agents completely digest as well as it rebuild their structure within the bacterial cell. So when this process is keep on going, at one fixed point, the number of phages or bacterial viruses within the bacteria gets increased. As a result, at the end of the cycle, the bacterial cell will be completely disrupted. So as a result of disruption, number of mature virions are released at a time. Virions are mature virus particles which are coming out from the bacterial cell. So this is said to be the lytic cycle, whereas this will not happen in lysogenic cycle because the bacterial DNA or the bacterial chromosome acclimatize the viral DNA which is inserted by the bacterial virus during the insertion process. Once the nucleic acid is inserted, the viral nucleic acid is strongly integrated with the bacterial chromosomal DNA. Fine. And this is the integration becomes so stable. The integration becomes so stable. So whenever the bacteria is subjected to adverse condition like excessive hydrogen peroxide treatment, excessive temperature treatment, excessive ultraviolet radiation, under these circumstances, the viral nucleic acid integrated into the chromosomal DNA may get excised, and this process is said to be induction. Induction of viral nucleic acid makes the virus to enter into the lytic cycle, what we discussed just before. So the minor difference between lytic cycle and lysogenic cycle is, in lytic cycle the host cell is completely destroyed or it is completely destructed, whereas in lysogenic cycle the cell is safe until it meets with the adverse condition. Once it met with adverse condition, the cell will definitely be disintegrated into small pieces due to the viral nucleic acid. Well, this is about transduction process, and the last one is said to be conjugation. Conjugation is a process which was explained by Lederberg and Tatum between two bacterial cells, and this is a very fundamental mechanism of genetic recombination between bacteria. Well, in conjugation process, what actually happens? So in other two methods, when in case of transduction as well as in the case of transformation, there is no need for the physical contact between two bacterial cells, whereas definitely in case of conjugation the physical contact is very important in this phenomena, right? What happens during the process of conjugation? So conjugation is a multi-step genetic recombination procedure. So the first step is determined by the proximity between two bacterial cells. Proximity, well, you can ask questions, sir, what is meant by proximity? Proximity here refers to how far the two cells are separated. How far the two cells are separated, right? So when the cells are close to each other, there is a greater chance for conjugation process. This is the fundamental and determining step in conjugation phenomena. Well, during the conjugation process, one cell is said to be donor, another one is said to be recipient. The donor cell is donated by the symbol F plus, whereas the the recipient cell, which is given as F minus. F stands for fertility factor. Fertility factor is defined as the character given by the new sort of DNA said to be the plasmid DNA. Those cells which contain this F plasmid are said to be donor, which is capable of donating its nucleic acid to the recipient cell. Which lacks the F plasmid becomes F minus, which means recipient, which is capable of accepting the genetic material donated by the donor cells. These are the difference between F plus and F minus. And F plus cells are capable of forming a special canal, and it is said to be pili. So pili, it is called as a conjugation canal, which connects the two bacterium which are in close contact with each other. So through the fertilization canal or through this conjugation canal, the genetic material between one bacterium and another bacterium is exchanged. Right. So during this process, the plasmid DNA of the donor cell becomes cleaved at one place, so that the double stranded DNA is converted into single stranded DNA. And this conversion takes place as a result of the formation of nick. Nick is defined as a small cleavage in the DNA. So this cleavage makes the double stranded DNA into single stranded DNA, and this single stranded DNA gradually enters into the another bacterial cell called as a recipient cell through the conjugation tube. And this conjugation tube is made up of a protein called pilin, and this is encoded by the plasmid DNA, which is the structural protein. Well, my dear students, this is about the video tutorial. Hope you have understand the basic concept of the genetic recombination or genetic exchange in bacteria. And please be remember, these three processes are fundamental and very basic before wherever you proceed into the higher studies. Thank you for listening. Thank you.